All right, so thank you all for coming back and continuing on with the lecture. So now we're going to talk about how much data to collect. All right, I talked a little bit earlier, and we're going to go into greater detail in later lectures on the difference between a population and a sample. But what we do, a population data set is what we're doing is we're trying to make inferences from the larger population. So one of the things that you remember we talked about the finer aspects of a research question. It has to be feasible. It's not feasible to sample the whole population in many situations, in many studies. And so what we do is we take a sample of patients from that population and we make inferences about that sample. We make inferences about the population from that sample of data. When you're talking about a population, and what we're talking about is we're looking at parameters. In a sample, we're looking at statistics. And so we measure the sample using statistics in order to draw inferences about the population and its parameters. Most research studies that are done in medicine use samples, and depending on how they collect that sample can depend on whether or not that sample is generalizable to the population of interest. So here's some examples of if we're thinking about how much data you might want to collect from populations or a sample. So I want you to do is pause the video, and I want you to think about, all right, which way would you do this? Would you do a sample, collect a sample for that one, or would you look at the population? All right. So the first question asks the average number of years of anesthesia experience of the CRNAs working in your hospital. Which would be the best way to answer that question? Would you do that with a sample or a population? Well, it might depend on how many CRNAs are at your at your hospital, but really you would probably look at the population. So say for example at my place, we had maybe 20 CRNAs. So we'd sample the whole population, in which case we're calculating parameters. What about a political poll? Well, that's really sampling, and there's a whole science behind that process. And I think we've even seen in recent elections that sometimes the political polls aren't always very accurate and representative of the population. What about the study of the rate of sleep apnea in parturients delivering at military hospitals over a 7-year period? It's a tricky one. Well, in fact, I did some research with my team a couple years ago, and we actually looked at the whole population of this. We actually were able to get the data set for all women who delivered at all military hospitals over a 7-year period. So we actually collected data on the entire population of interest for that period. But we could have done a sample where we randomly sampled women in each year group, and then in which case we would have been calculating statistics, not parameters. Here's some other examples. You randomly select 20 nurses at your hospital to complete a job satisfaction survey. The sample would be the 20 nurses. Your population that you're making inferences or drawing conclusions about are all nurses at your hospital. How about the ANA randomly selects 5,000 members and sends them an electronic survey about annual compensation and benefits? Well, we know that there's closer to 50,000 ANA CRNAs, and over 90% of them, I think over 95% are ANA members. Well, it's really a sample of the larger population of CRNAs. You hand out a survey to every CRNA in your anesthesia department. Well, that's really just a population. Maybe you only have 10 of you in your 10 partners in your group. So you've sampled. So you've collected data from the entire population. So a few more terms to know in terms of survey research is response rate and completion rate. So the response rate is the actual number of participants divided by the number surveyed. And so you send out a survey to 1,000 CRNAs and only 200 volunteer to complete it. So your response rate is 20. Now you may have 20%. However, not all of them complete all the questions on the survey. Maybe you had 30 questions and they only completed, maybe only 50% completed the whole survey. So that's what your completion rate is, the number of completed surveys divided by the number of respondents who entered the survey. So here's an example. Identify the population, sample, and variable in this particular example. So a DNA-pCRNA student at Texas Wesleyan analyzed data from the MBCRNA database on 250 randomly selected sRNAs who took the NCE exam. His goal was to determine first-time pass rate. So the population that you're interested in are all CRNAs who took the NCE in 2016. The sample may be 250 randomly selected sRNAs from across the country. A variable would be the NCE first time pass rate in 2016. Now there's some issues you have to consider with sampling. Is the sample size can be too small, so you have a higher chance of finding the result even if there is one, so the study can be underpowered. That's the most common thing that we see. Or pretty rare, but I have read a few studies where the sample size is so large that you may find spurious findings that they may be statistically significant, but they're clinically irrelevant. It can be time consuming and costly to have large sample size. So we really have to balance the risk benefit when we're choosing. We have to base that on evidence, and so that's why many times we do power analysis to make sure that we're being most efficient with our scarce resources. So organizing the data. We already talked about a little bit of this earlier, how we might organize a data set. The first thing that you're going to do is you'll develop your data collection tools or identify instruments for collecting your data. You would develop a database and a code book. After you're done collecting your data, or ideally throughout the course of the study, you or your research assistant or whoever, your partner, is entering the data as they go, because I can tell you, try and do this all at the very end, it's a lot of work and you sometimes will make mistakes. You clean your data set, meaning you got to go through and identify. You'll run some descriptive statistics. You'll look at it to see if maybe you, instead of typing a sorry, let me go back here. So maybe instead of you go through and run some frequencies and you look at the data and you look at gender and you run a frequency distribution, and maybe you found you accidentally typed a couple threes in there. Well, you only coded them as a 1 or 2, and so you could identify that when you're going through and cleaning it. So then what we're doing is we run some simple initial descriptive statistics to help us look at the data or frequencies, and there's a lot of different ways that we can do that, and we'll talk about that later. You can have a frequency table for gender. Here you might have a frequency table for stop bang category right here. You could also go through and calculate means and medians for your other variables as well. So like we could even calculate the average stop bang score or the average or the mean weight, and we can look at the variability in that. So then maybe we're going through and presenting the data. So then maybe we then turn around after looking at the frequencies of data. Sometimes that's not an easy way to look at data. So then we can look at presenting it in bar graphs, which are easier for the reader to interpret. So there we ended up taking our frequency tables and converting them into tables. And you can see that each variable that we collected here had a graph associated with it. Many software like SPSS will automatically do this for you. So it's really nice when you're going through and looking at your data that you have. Then you want to go through and describe your data what you collected. In which case we're using graphs and tables are the easiest way to present all your data. Later on in you guys' program where you're going to work on your final papers for graduation, you're going to be synthesizing a lot of literature into a summary table. You don't want to type all those numbers into a paragraph. That's too difficult to read. Nobody's going to read it. So you have to be able to summarize that into tables. But in research, we do the same thing. Is that we use tables and figures to really use the efficient use of that journal article space, and it's easier to follow. And the reason we use statistics so we can use graph and tables to present the data, but it doesn't really help us many times answer our hypotheses or test our hypotheses. So then, in which case, we want to run statistical analysis so that we can make inferences about the population from which we sampled our data from. And we can use precision measures, which means we're trying to get a little bit more precise so that we can make inferences about the population, assuming that the sample is similar with the population. And so example precision measures may be the mean, the median, the mode, the degree of variance, the standard deviation, or the proportion. So what we may be doing is we take a random sample, we calculate some statistics, and we make inferences about the population that we're interested in. Doesn't always work, but many times it does. Another ways is we may be looking at, is there association, relationship, or correlation or interaction between two variables? Meaning, are they, when we do this, we're talking about, is there dependence or independence? When we say there's dependence, that means that there's a relationship that one depends on the other. For example, if you look at correlations, a change in one results in a change in other, either positive correlation, meaning as one value goes up, the other one goes up, or negative, as one goes up, the other goes down. Or here, if you were to run a correlation, you don't see there's no relationship between these two variables. So a change in one variable is not associated with a change in another. I'm going to pause there, and then you guys can continue watching the next video.